All right, I am Sudhansu, and I am working with the PeanutBase, and I changed the topic slightly differently, and I apologize for that because the tool is not completely ready or rather integrated yet, but I will give enough information what we will be getting out of that tool along with other tools at PeanutBase. Oh, I don't. Okay. So PeanutBase, right from the beginning in 2013, has been supported by the Peanut Research Foundation and guided by the Peanut Genome initiatives. We were given the responsibility to serve as a portal along with the genetics data, the genomics data coming from the diploid ancestral genomes, and later the cultivated peanut genome, the hypogaea genome, and very soon there will be a few more diploid genomes available at PeanutBase, and these are our main activities. And besides that, sorry about that. Besides that, as major data types came from the research community, PeanutBase also evolved to accommodate all those data, both for storing in this web portal as well as for sharing with the community. And in addition, we also are involved in developing tools to explore those data, particularly visualization tools. And for example, we have genetic markers from various mapping studies, and also from the genotyping chip. From the green accession, we have quite a bit of germplasm data along with their images and their evaluation data we have compiled, and they are accessible not just as data in tables as well as in a map, GIS map context. One can localize the collections and the traits in a world map which is very interactive. We have genetic data, of course, quite a few QTL data sets. We have an expression atlas. And this has been our major, motivation. Can I get rid of this strip at the bottom, which is affecting the visibility? How do I do that? Okay, sorry. So whenever data or any data type comes from the community, we gear ourselves towards setting tools to both store and analysis if necessary, and visualization tools to explore such data sets. Oh, I'm sorry, I'm having trouble navigating. Okay, got it. So when we had genotyping data sets from the Peanut Innovation Lab projects, the African breeding lines, we adapted the GigWord tool to store those data. This was a tool that was very successful in helping us sharing with the community and a few simple explorations too. But we also felt the limitation of data, particularly the graphic visualization. So PeanutBase got involved with Steve Cannon's group and developed a tool for the genotype comparison visualization tool called GCVIT, which can compare different genotypes and particularly in the context of where these genotypes differ and where they match in a genomic context. And here I have shown an example and along a few chromosomes. And I have three columns here in red. These represent the SNP data coming from the cultivated peanuts, and the rest colored, differently colored, 4 columns coming from different land races. And wherever there is a white area, this shows there is similarity in the haplotype feature between these columns that represent the genetic lines. And if there is a color, it shows there is difference. And one can use to compare the parents versus its derived lines. One can compare, if necessary, the breeding during backcrossing, the progress of that. But more importantly, one can also check regions where integration has taken place in the cultivated genome coming from different land races or even from wild diploid types. And I know some people have started with this to narrow down from these bird's eye view features to reach the genomic region where they can see what are the genomic features, particularly the markers, molecular markers they can use. And we hope this becomes useful to the community later. And this is a new feature which we had advertised before, but now it's fully functional at PeanutBase. This is Arachis Fennel. This is our tool to explore phenotypic data, all the trait measurements. And particularly, like I have shown here, one can see the distribution of a trait in that collection or in that population. And one can see whether it's symmetrically distributed with normal distribution, or if there is a skew among the various lines in that collection. And on the fly, if we have the data stored in PeanutBase, one can see the scatter plot of a trait. How is the distribution of all the measurement values from that phenotype? And one can also plot the correlation between any two phenotypes and see the strength of correlation. Particularly if it's a selection experiment, sometimes readers might like to know if two features are highly correlated, then they can make some selection program decisions based on them, which one to emphasize more, or if there is a negative correlation, or if they are independent of more or less independent of each other. And we have only the minicore data represented in the phenotype so far, and our aim is to popularize this feature. The more there is the phenotype data in this tool, the more useful it becomes to explore among various collections. And I encourage all those labs who have this kind of data to contact us, and we will help to accommodate those data in this new tool. And here, as soon as GWAS data became available with the peanut researchers, we developed a tool, and it's with an example from the NAM GWAS data from the 2 NAM populations. And this GBrowse, which is a modification, local modification of the GBrowse tool. And beyond the Manhattan plot, this is an interactive plot. One can click and go to a very narrow area. For example, here with respect to pod weight and seed weight, and here I have shown I can focus on any of these and look at, from the genetics point of view, look at the genomic landscape in that area, particularly the synteny features. And also these colors represent the gene family of those in the genomic, narrow genomic region. And based on the microsynteny data that we have for each of these genomes, I can also traverse to another species. And here I have shown with to soybean and go do the reverse travel from the genomic landscape in that region back to the QTL data, which in this case happens to be another pod trait in the same nearby region. And I initially wanted to emphasize the PeanutMine. The tool is ready, all its parts, but we have had some difficulty transferring data from one technology stack to this one. But I will just show that this tool is based on the PeanutMine, which has been adopted by many like fly-based, nematode-based, Arabidopsis, and besides the regular search features of various data types, it has the list analysis facility, which we had lacked in PeanutBase before. You can take a list of genes and do various kind of things like get all the FASTA sequences for those genes, or get the mRNA CD sequences for those, or find out the corresponding transcripts or the protein sequences. Instead of going by one feature by another, you can take the whole list and get all those data together. And the strength of this tool lies the way different data types are connected. I cannot demo this, but what I can do is show you this.
You have about 2 minutes, Sudhansu.
All right, I will try to finish in 2 minutes. This can be reached from the homepage of PeanutBase. One can search and give a list of genes here and analyze, do some, you can take gene names, protein IDs, QTL trait names, or GWAS features. And with the genomic features, we have ready-made templates which can do the query from the genomic region, given the coordinates, to what are the genes present in that region, or from genes to the Go terms or back. If we have Go terms and you are interested in what are the genes in peanut, you can get those too, and you can go given a gene description, like let's say chlorophyll biosynthesis, one can immediately go and get all the genes and narrow it down. And here we have a feature. I have looked for chlorophyll biosynthesis genes, and we have about 10,000 genes, but one can restrict those just to one of those cultivated peanut, or from the diploid species, or by the Go term. And these features will be more enhanced as we integrate more data here. And this is one of those. If I go by a genomic region, I can also extract all the genetic markers there and keep them in a defined list to be analyzed further. And I have already shown that. I'm now going back. And okay. Now we have peanut core collection genotyping project data, a few of them, phenotype data, GWAS data, and more QTL data will be inserted there. We have also taken care of pathway data in the PeanutMine as well, along with various visualization of genomic data using a few recently developed tools with a new sequence search feature called Sequence Server. And most of our emphasis will be providing the stewardship of the genomes, new genomes that would be coming, and efficient, streamlined workflow to integrate the genetic and the genomic data, and particularly the link these features. The collaboration is with these two groups, the Stephen Cannon's groups in USDA in Ames, Iowa, and Andrew Farmer's group in NCGR in Santa Fe. And along with these other projects we collaborate from. And the main help comes from Stephen Cannon's group and Andrew Farmer, who very generously share their expertise and personnel to develop the PeanutBase. And thank you very much. And I solicit data, whatever data type is there, please contact us to house in PeanutBase. Yeah, thank you.